Good morning, students. Students, am I audible?
Good morning, sir.
Very good morning. Am I audible to everyone?
Yes, sir.
Yes, sir.
Right. Very good. So yesterday we discussed about the unit of life, right? That's a part of cell division. And let us continue. Microscopy. Yesterday I started the teaching of microscope. We have discussed several types of microscope, right? And let us continue some types of microscope, right? That is very interesting topic. So please pay your attention. As we have discussed, microscope is nothing but it is an instrument that is used to see an image in maximum size, right? That is called a microscope. A microscope, it is an instrument that can be used to view an image, microscopic structure. When we place under microscope, we can see big size, right? That is called a microscope. Micro, the term micro means small. Micro, already we have discussed, right? And the small size of an object can be seen in maximum size, right? That is called a microscope. So we have discussed several types of microscope, right? And once again, let me point out, they are a simple compound microscope and bright field electron microscope. Here, already we have discussed several types, and let us continue some more types, right? The first one is a bright field microscope, and second one is a electron microscope. The first category is a bright field. Bright field microscope. And second type is
Electron microscope.
electron microscope. Very good. The second category is the electron microscope. There is a difference between bright field and electron microscope. Bright field, as the name suggests, bright field microscope that produces light which passes through the specimen when we place under the microscope. That is called a bright field microscope. Here, a source of light is used artificially when we give any microscopic slide under the microscope. We can use some amount of light artificially. That is called a bright field microscope. Now, light here is focused on the specimen. When we place on the stage of microscope, hence we can see the ultrastructure of an object, ultrastructure of any object when we place under the microscope. So that is called a bright field microscope. Whereas electron microscope means it is a kind of microscope in which electrons are utilized for seeing of an image when we place under the microscope. That is called electron microscope. Simply I can say, in bright field microscope, light is utilized for viewing as a structure on the stage of your microscope. Whereas electron microscope means a beam of electron or the electron sources are utilized on seeing the ultra structure of any object, right? That is called a electron microscope. Okay, and the bright field, one more I want to mention, the bright field microscope. It is an instrument which is used to see an object. It may be 5x. 5x means nothing but the magnifications or resolving power is a 5x means x indicates a time or size of an image. Simply, when we place any object under the microscope, now this is an object, right? When we place any object, so it is a x means it is a normal size of an object is said to be x. Here, a normal size of an object can be viewed, can be increased by 5 times greater than the normal size of an image, right? That is called x. So here, the 5 times of x, it is when we consider it is an object, it can be increased its size of 5 times greater than the normal size. So that is called 5x. Here, the bright field microscope can be used to view, to see, to increase 5 times greater than the normal size of an object. Right? That is simply I can say 5 times increased of its original size. That is called the 5x. And sometimes it may be increased like a 10x. 10x means 10 times greater than the its original size of an object. That is called 10x. And one more term, right? I can mention that is a 45x. So 45x means nothing but 45 times of its original size can be increased when we place under this bright field microscope. And last one more category, that is 100 times. Just imagine it is a 100x. That means a normal object can be increased its size 100 times. That is called a 100x. That is indicated by 100x. No, x is nothing but it is a normal size of an object when we place under the microscope. Right? So that is called a 5x, 10x, 45x and 100x. So depending upon the size of an image, here we use different kinds of lens system. In order to view 5 times greater than its normal size, we can use a slide or the lens system is a 5x lens. So that's why I mentioned there are different size, different categories of lens system. They are 5x lens that is used to view 5 times greater than its normal position. It is a 5x lens. Whereas a 10x lens, it's a type of lens that can be used to view 10 times greater than its normal size. That is a 10x. Whereas 45x means it is a kind of lens which is fixed on the objective lens system, and that can be increased to 45 times an object. Whereas last lens system, that is a 100x lens. I'm talking about different types of lens system. It is a 100x lens. It is a kind of lens which can be used to increase 100 times of its original size. When we place any object under the microscope on a slide. So that is called 100x. So here, all the various types of lens system are fixed or rotated on the objective lens system. That is called a 5x, 10x, 45x and 100x. Now here it can be used commonly and routinely, right? Sometimes when if you want to see 5 times greater than that of an object, you can use a 5x. Or if you want to increase the size of the objective when we place under the microscope, you can use a 10x. Or if you want to see a maximum size of an object by using bright field microscope, you can use 100x. No, 100x is nothing but it is a lens system that can be used to increase an object is 100 times greater than that of its original size, right? So that is called 5x, 10x, 45x, and 100x. That's why I mentioned this is a bright field microscope. It consists of various types of lens system, right? So this is a simply something but of a bright field microscope, and what is the use of this microscope, right? Actually, this is a bright field microscope, right? That is used to see the structure of tissues and tissue system. Tissues and tissue system and its types. You know very well in plants there are several types of tissues. Simple tissue and complex tissue, meristematic tissue. So in order to know the fine structure of tissues, we can use a bright field microscope. If you keep any type of tissue under the microscope, you can find. So for example, let me find on. Now this is a simply parenchymatous tissue. Parenchyma. It is a simple tissue. The ultrastructure of parenchyma tissue can be seen under the bright field microscope. And one more tissue I want to mention, that is a collenchyma tissue. But next we are going to study in detail, right, about the tissue. What is tissue and what is tissue system? Tissue is nothing but it's a group of cells. It's known as a tissue. I repeat it again. What is tissue? A group of cells is called a tissue. It may be parenchyma or collenchyma, and one more category, sclerenchyma. So the ultrastructure of tissues can be seen under the bright field microscope. As I said, this is a bright field microscope that utilizes light, right? Actually, light is emitted from the light, which is emitted from the light, which is emitted from the LED. Light emitting diode, LED bulb. Actually, bright field microscope that consists of light system, namely LED light that passes. When it passes the light on the specimen, the image is captured, right, by objective lens and which is uplifted to the eyepiece lens. So talking about microscope, I'd like to mention this is a bright field microscope, right? Just see, you can understand. Dear students, now this is a bright field microscope where you see different lens system, right? As I said, it consists of several types of lens system, right? Here, this is a right example of right diagram for bright field microscope. Now, I'm talking about light. This is a light, right? Actually, this is a light which emits from the LED light. LED bulb is there. Actually, this LED bulb is nothing but a light emitting diode. Light emitting diode. That is the abbreviation of LED. Light emitting diode. Or it is also known simply, instead of LED, sometimes we can use a CFL, right? CFL. That is a bulb that can be also used, CFL, right? Generally, just imagine LED bulb is there in the bottom of bright field microscope. Actually, this bright field microscope, it consists of mainly three parts. If you had to understand, the first one is the body of the microscope. Now, this is the body, right? It is the body of the microscope, and it consists of the eyepiece lens. It is known as simply eyepiece lens. Eyepiece lens, right? This eyepiece lens that is very closer to eye, and you can see the objective lens. Now, this is an objective lens, right? It is an objective lens. Actually, this objective lens is a lens which is very close to the specimen. Now, this is called a specimen. It is a specimen. So you can take any type of specimen, whether parenchyma or collenchyma or sclerenchyma, specimen. So when we place a specimen on this is a stage, very fine. It is a stage. It is a stage. The stage is a platform on which the adjustable microscopic slide can be kept on that, right? That is a stage. Actually, stage is a platform which carries out a specimen lying on the microscopic slide, which is fixed on the stage of a microscope. And just very next to the specimen, you can see diaphragm. Actually, this is called a diaphragm. So, diaphragm is a place that allows the light system. Actually, the light system which emits from the LED bulb, right? This is a light system for LED. This bulb which emits light system, and this light system can be allowed by diaphragm, right? So either it increases or decreases depending upon the specimen that we take when we place under the microscope. Understand? So if you want to increase the brightness of the specimen, just we can adjust this diaphragm which is found just below the stage or platform of the microscope, right? Okay, here this LED is emitting continuously light. The light which passes to the specimen through the diaphragm. So when the light passes on the specimen, the ultra structure can be seen very clearly, and this image can be captured by objective lens and which is passed towards eyepiece lens, and this eyepiece lens which is also captured, right? So that is called simply you can say magnification. So here the magnification, there are two types of magnification. Is nothing but it is an optical increase. A size of an image is called as magnification. Dear student, you have to understand what is magnification. It is nothing but it is an optical increase in size of an image. Yesterday we discussed about magnification. Yesterday we discussed about magnification. Now magnification is nothing but it is an optical increase. It is an optical increase in size of an image. The size of an image. Right? Magnification is nothing but it is an optical increase in size of an image when we place under the microscope. Now, this magnification can be calculated by some formula. So, therefore, magnification is always equal to, right? Magnification is it can be calculated by using a formula that is a size of an image. Therefore, magnification M is equal to, right, the size of an image. Size of an image, say, with the microscope with a microscope by a size of an image, by size of an image. See with the height. So that is called a magnification, right? The magnification can be calculated by using a formula, that is the size of an image is seen with the under microscope, by the size of an image is seen with our naked eye. That is the ratio. So the ratio between size of image which is seen under the microscope by the size of an image is seen with our naked eye. That ratio, that is called the magnification. That's why I mentioned that magnification simply it is an optical increase in size of an image. That is called a magnification, right? So here we're talking about magnification. There are two types of magnification, right? The one is primary magnification. Magnification. As I said, magnification, it is nothing but it is a size of an image, right? It is an optical size of an image under the microscope, that is called the magnification. Now, it is a two types. They are a primary magnification and the secondary magnification. Primary magnification, and the second one is a secondary magnification. What is primary magnification? Actually, primary magnification is nothing. It is an optical increase in size of an image with high piece lengths. So simply I can say, the real image, which is seen inverted in condition, right? The real image which is seen, or it is in a real image is seen with the high piece lengths. The high piece lengths. The high piece lengths. Real image is seen with the high piece lengths. That is a primary magnification, right? Or simply you can say the image is seen with the real image is seen with the high piece length. That is a primary magnification. Whereas the secondary magnification is nothing but an objective. Simply you can say an objective. And a real image or virtual image is seen. Virtual image seen near the objective lens, So that is called a secondary magnification, right? So simply an image is seen with our naked eye by using of eyepiece, that is a primary. Whereas a secondary magnification, it is a virtual image is seen objective lens, right? In inverted position, in inverted condition. So that is called the primary and secondary magnification. So please understand, we have discussed there are two types of magnification. One is a primary and another one is a secondary magnification, right? Next we pass on to another type of microscope, that is the electron microscope, right? Dear student, please understand, we have discussed simply the only one microscope, that is a bright field microscope, right? Next we pass on to another type, that is a electron microscope. Dear students, we are going to discuss about simply electron microscope. Electron microscope. Actually, electron microscope, it is an instrument that can be used to increase an objective, right? As 1 to 3 lakhs time. 1 to 3 lakhs time. That means an objective can be increased 1 to 3 lakhs of its original size. So simply it shows a great range of variation in the structure of an objective when we place under the microscope, right? So that is the electron microscope, right? Actually, this electron microscope, it was first introduced by Ernest Ruska. It was first introduced by Ernest Ruska. Ernest Ruska in the year of 1931 was the first man to be introduced electron microscope. I repeat once again, electron microscope, it is an instrument that can be also used to view the ultra structure of plant cell or cell. Simply, electron microscope, it is an instrument that can be used to view, to see, to find the fine structure of or ultra structure of cell. Ultra structure of cell and cell organelles. In first case, we discussed it is an instrument, bright field microscope, that can be used to view the fine structure of tissues. Whereas here, it is an instrument which is also used in laboratory to view the fine structure of a cell or ultra structure of cell and its cell organelles like mitochondria, nucleus, Golgi apparatus, endoplasmic, etc. So these cell organelles can be seen very clearly. Not only that, the fine structure of cell also seen when we place this specimen cell as a specimen under the electron microscope, right? So this was first introduced by Ernest Ruska in the year of 1931. And later on, there are two people who jointly developed such an instrument that is called electron microscope, right? Namely, there are two people who jointly developed this instrument. Binning and Roher. Binning and Roher in the year of 1981. These two scientists who developed this electron microscope, right? Actually, Binning and Roher, they are the people who developed such an electron microscope, right? Now, what is the use of electron microscope? The electron microscope, it is an instrument that can be used to view, to see the fine structure of cell and its cell organelle. Yes, student, could you tell me who introduced the first electron microscope? Could you tell me the name of the scientist?
Ernest Ruska.
Very good. Could you mention the year?
1931.
Okay. What can be seen under the electron microscope?
Ultra structure of
cell and cell organelle.
Cell and its cell organelle. Yesterday we discussed about cell biology. Yesterday we started cell biology. It's a branch of biological science that deals with the study of fine structure of cell and the cell organ that we have discussed. Such a fine structure can be studied very detailed by using of electron microscope, right? So far we have discussed the simple compound microscope and bright field microscope. It is very interesting microscope, important microscope, right? Due to discovery of such a microscope, the branch of science, that is cell biology, has been developed rapidly due to invention of electron microscope. Because the detailed study of cell and its cell organelle, from that molecular biology has been also originated very rapidly, right? That is simply discovery, a history of this such a microscope, right? So, before the 18th century, the simple microscope has been and the compound microscope has been only used by the scientists. But later on, right in 19th century, this electron microscope, so it has contributed many aspects to science. So please understand. Okay, talking about electron microscope, simply the electrons are reacted with one another on the specimen when we place under the microscope. Hence, the new image is captured by passing of electrons on the specimen. So directly we can detect the nature of exactly the fine structure of cell and cell organelle on the fluorescent screen. Actually, here there are some components of electron microscope, right? Just I'm going to tell you one by one. And before that, the microscope, it is of simply, what are the major components of this electron microscope? Simply, this electron microscope, it consists of electron generating system. That is the first component. As I said, it consists of the electron generating system. Electron generating system that plays a major role, right, in the development of fine structure of a cell, right? Because it generates continuously a beam of electron which are passing on the specimen, right? So there is a system that emits continuously electron, right? These the electrons are falling on the specimen. So when the electron flows continuously from the electron generating system, what will happen? The electrons are reacted with one another. So hence there are different radiations also emitted from the specimen. So hence the visual image can be captured by the radiation emitted by the electron when the electrons are scattered. When the electrons are scattered or when the electrons are reacted with each other, when we pass on the specimen from the electron generating system to the specimen. So there is a new image or the fine structure can be captured by electron detector or image detector, which is on the bottom of the electron microscope, right? So this is a simply electron generating system. And one more, it also consists of electron condenser. It also possesses electron condenser. This electron condenser, which controls the beam or the size or the amount of electron, the quantity of electron can be measured, can be controlled by, that is electron condenser. So it allows the passing of the amount of electrons emitted from the electron generating system, and the amount of electrons are controlled by electron condenser, and it allows a particular amount of electrons are emitted or allowing on the specimen. So simply it controls the path of electrons, right, which are going to capture, which are going to fall on the, which are going to fall on the specimen when we place under the microscope, right? The next, tube lens. It also consists of a tube lens, right? It also consists of tube lens and detector. Finally, you can say detector or screen. The screen is there. So these are all the components of electron microscope. Okay, talking about the electron microscope, it is of two types. They are TEM and SEM. Simply, the electron microscope, it is of two types, right? They are TEM and SEM. First of all, let me take a TEM electron microscope. TEM electron microscope. It is a microscope which is also used for a brain, right? And produces, it shows, it makes it 2D. So TEM is nothing but a transmission electron microscope. TEM is nothing but transmission. Transmission, E stands for electron. Electron, M stands for microscope. The term is nothing but transmission electron microscope. This microscope which increases its size of a magnification value, right? 1 to 3 lakhs times. 1 to 3 lakhs times of an image, right? When we place, this is an image, right? The fine structure, it is a microscopic slide, right? When we place any image, the image can be seen or increased by its original size 1 to 3 lakh times. 1 to 3 lakhs times. The magnification value. Magnification is nothing but it is an optical increase of an image, right? So this optical increase can be 1 to 3 lakhs times greater than that of a normal size of an image when we place under the TEM microscope or transmission electron microscope, right? Okay, and it also, and its resolving power, the resolving power value is 2 to 10 angstrom inside, right? The resolving power. Resolving power, it is the ability of a lens system to resolve the natural size of the image, right? That is a resolving power. Resolving power is nothing, it is the ability of a lens system which are utilized, which are fixed on the microscope, right? To resolve, to show an image. That is the resolving power, right? So the resolving power is a 2 to 10 angstrom inside. That is very important, right? Now, what is the use of this term electron microscope? It also consists of simply, before that, it consists of electron generating system and the condenser and projector or the screening, etc. Right? So I want to mention here what are the components of electron microscope. The first one is, as I said, the electron generating system, and it also consists of one more, electron condenser. EC, electron condenser, right? Electron condenser that regulates the amount of electrons needed for viewing or passing on the objective specimen, right? And it also consists of a tube, right? Tube lens. And one more, the final we can say projector or screen. It is going to project, right? So simply when we place any object, right? This is I would like to show you. Dear students, do you want to see the structure of electron microscope, TEM microscope?
Yes, sir.
Do you want to see?
Yes, sir.
Right. Please wait, let me show you. Let me show you. Now, this is a TEM electron microscope. It is an instrument, right? It consists of, as I said, it consists of, there are different components of electron. One is electron generating system. Now, this is electron generating system, and the electron condenser is there. And this electron generating system passes continuously electrons. Actually, here the specimen is there. Now, this is a specimen. I like to mention in red color. This is a specimen where it is a platform where it is a stage where we place a specimen, right? So, when we place any plant cell or animal cell, right, or bacterial cell under the microscope, you can see the electron generating system that emits continuously electron. The electrons are regulated by the electron condenser. And finally, just bottom of the electron condenser, you can see a tube lens. This tube lens which allows this electron, and this electron are reacted on the specimen, or the continuously coming out electron from the electron generating system. They are reacted with each other, and it forms different kinds of electron. They are auger electron and secondary electron and backward. So all these electrons are reacted and it forms a different radiation. Now this radiation which will capture an image which is placed on the microscope. Now this image can be detected on the scene on the projector. That is called a projector. Actually, this is a projector. It is an instrument. It is also seen just bottom of the electron microscope, particularly you can say TEM microscope, which views the original size of the structure of the microscope, original structure of an object, whatever we place. Just imagine now this is a plant cell, right? Just imagine it is a plant cell. So this is a plant cell. The ultrastructure of plant cell can be viewed very clearly, right? By using this microscope, right? It is a plant cell. Not only this plant cell can be seen, but also a bacterial cell and the viral cell, mycoplasma, and the cell organelles. Not only cell can be seen under the TEM microscope, but also we can see virus. Virus can be seen. The first structure is a virus. Virus. Already we have discussed about the different kinds of viruses, right? T4 bacteriophage, tobacco mosaic virus, TB virus. So the fine structure of a virus can be seen under the microscope by using electron microscope or TEM microscope. And not only that, mycoplasma also seen. Mycoplasma can be seen under electron microscope. And the cell organelles. The cell organelles can be also seen under TEM microscope or transmission electron microscope. So this is something about a TEM microscope. I repeat once again, TEM microscope, it is an instrument that can be used to view 1 to 3 lakhs times. Its normal size of an image can be increased, right? So the magnification value may be 1 to 3 lakhs times, and it shows a 2-dimensional size, right? So that is the nature of a TEM microscope. Whereas, and when we use a SEM, the SEM is nothing but a scanning electron microscope. SEM. It is a type of electron microscope that provides, that increases, that shows a 3D structure. SEM is nothing but scanning electron microscope. Scanning. Scanning, E stands for electron. M stands for microscope. Hence, SEM stands for, S stands for scanning, E stands for electron, and M stands for microscope. So it is a type of electron microscope, and that is used to increase its time, right? Its magnification value is 1 to 2 lakhs time. But it provides a 3D structure, 3-dimensional structure of an image. That means 3D view. A structure can be viewed in 3-dimensional structure. Any object, when we place under the SEM microscope or scanning electron microscope, the image can be seen as a 3D structure, 3-dimensional view. An object can be viewed as a 3D structure. That is a special characteristic which is a SEM. Whereas TEM, while discussing TEM, it shows only 2D structure, whereas the SEM, which provides a 3D structure of an image when we place under the microscope. But as compared with TEM, it is the lower than of TEM, because it is very low here. Because the magnification value is simply 1 to 2 lakhs, whereas in TEM there will be 1 to 3 lakhs times of magnification value. Talking about its resolving power is simply resolving power is a 5 to 20 a nanometer, right? I mean, Armstrong image. Resolving power is a 5 to 10. So this is a question that you can expect in an examination. Dear student, please understand resolving power is 5 to 20 nanometer, but its magnification is 1 to 2 lakhs of an objective. So here also when the electrons are kind of reactor. So this is a dear student, would you like to see? My dear students, would you like to see the structure of TEM microscope? Some
Yes, sir.
Yes, sir.
microscope. This is a simply I have collected for you. And this is a microscope. It's like a mission, right? It also consists of electron generating system. Now this is a electron generating system, EGS, and here you can see electron condenser and tubulets is there, right? And an objective is now this instrument, right? It also possesses simply a stage or where, and the electrons are reacted, hence it forms different kinds of electron radiation. When the electrons are reacted on falling or passing on the specimen continuously, the electrons are emitted from the electron generating system, and each electrons are reacted with one another and to form a different radiation. Such radiations are nothing but they are the, you can say, auger electrons by emission of radiation emitted from the specimen. Hence we can get auger electron, right? This is the auger electron. And the second one is the secondary electrons, right? And the third one is a back scattered electron. Back scattered electron. Because the electrons are reacted and it forms a different radiation such as other electron. So this is a question mark. Dear student, please understand that. In which of the following electron microscope, when the electrons are reacted, the emission of radiation is in the form of dust? That is the question, right? Dear student, please understand this is very important area in examination point of view. When in which of the following electron microscope, and the electrons are reacted with each other, it forms a different kinds of radiation, right? Such as auger electron, secondary, and backscattered electron. Please name the electron microscope.
Backscattered.
Scanning electron microscope, right? Okay, when we place an object under the scanning electron microscope, the object can be, or an image can be seen as a three-dimensional view. Now, this is a three-dimensional view. So, this is an image which is captured by the detector on the or seen on the fluorescent screen. So, the image can be projected on the computer. Right here, you can see an image can be seen as it is, right? By scattering of electron. Actually, this electron generating system and continuously passes electrons from the EGS and regulated by EGC and falling on the specimen when we place under the microscope. And the electrons are reacted after falling on the specimen are reacted with each other and it forms a different radiation, auger and the secondary back scattered electron. Right. Finally, the image is captured by the radiation and seen on the screen, fluorescent screen. So this is a simply something put out for SEM, right? A scanning electron microscope. Dear student, we have discussed there are simply two microscopes today. They are light field or bright field microscope and electron microscope. Any doubt regarding this topic? My dear students, did you understand today?
Yes, sir.
Yes, sir.
Right. Could you mention the different size of the lens system that we are using in bright field microscope?
5x,
5x,
10x,
10x,
45x,
45x, maximum 100x.
very good. Right. 5x, what do you mean by 5x? How did you understand about 5x?
Size of
greater than
5 times of x.
5
times.
times of an image can be increased. Right. What do you understand about 10x?
10 times
10x
larger than the small
image
size.
can be increased. That is a 10x. Actually, what are they? 5x, 10x, 45x, and 100x.
size.
The size of the
Size of the bright field microscope.
actually 5x it is a lens, 10x lens, 45x lens, 100x lens. So that's why I mentioned there are different lenses that we are using in bright field microscope. Okay, could you mention the source of light emitting from the bright field microscope?
Light emitting diode.
Emitting diode, LED.
Diode. Simply you could say LED.
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 Bulb. Very good. Now this is a microscope, right? Sure, you can see. Okay, could you mention the LED bulb where it is fitted? Whether bottom of the microscope or top of the microscope?
Bottom of the microscope.
Bottom of the microscope. And could you mention a high-piece lens where it is fitted?
At the top of the microscope.
Top of the microscope.
Very good. Okay, I'm seeing the microscope as we have discussed. There are different kinds of lens system. They are 10x and 45x.
45x, 5x, up to 100x.
These lens, where are they fixed? Where are they fixed?
Stage.
Sir, stage.
Objective of the board.
Objective in
of the microscope.
Actually, objective lens is a lens which is very close to the
Object of particle.
and microscope, any object, right? The lens is very close. This is a objective lens.
Objective lens.
10x and 45x and 100x
100x.
can be fixed on the objective lens system. Understand? Very good, man. Right. What do you know about magnification? Could you understand about magnification?
An optical increase in size
optical increase in size of an image.
Very good, ma'am. And there is an optical increase in
increase in size of an image.
Increase in size of the image.
Right. Very good. Could you mention the types of magnification?
Primary
And secondary.
Could you mention the people who developed the electron microscope?
Ernest Ruska in 1931. Ultrastructure of cell organelles.
Who mentioned the types of electron microscope?
So two types.
Two types.
Types.
Electron microscope,
Scanning
electron microscope.
electron microscope.
Okay. Talking about TEM, transmission electron microscope, can be seen only the structure of
2D.
Two dimensions.
Two dimensions.
Image.
whereas in SEM can be seen.
Three
Three-dimensional.
dimensions.
Very good. Will you mention the radiations emitted from the electrons?
Agar electron.
Agar electron.
Agar electron, black scattered electron.
Black scattered electron.
electrons.
So thank you for joining, my dear students. Thank you. And
Thank you,
prepare-
sir.
Thank you, sir.
Definitely you can expect some question, right? So thank you for joining.
Yes, sir.
Thank you for joining, my dear students. And don't waste our time, please write and try to prepare all the content what we are going to teach on online, right? home assignment. Please take down the home assignment. Some question I'd like to give.